So, good morning, everybody. Buenos dias. Bon dia. Welcome to our symposium on adaptation to abiotic stresses in farm animals. Today we have people from all over the world. We're going to look at how animals adapt to abiotic stress, looking from stressors from questions of air temperature, what we can expect from global climate change, how this can be expected to affect the productivity of our farm animal production systems, how researchers all over the world are looking towards increased selection for production traits, how these stressors can reduce fitness, how highly selected animals are less able to respond. We're also looking at how animals adapted to abiotic stressors in specific environments could also help commercial production systems to elucidate pathways for adaptation and aid in the mitigation of stress. Typical adaptations that we're going to be seeing are those that are related to physical characteristics such as hair, wool, fiber, morphological characteristics such as body size and shape, behavioral, biochemical, physiological changes that can occur, as well as genetic responses to these changes in these stressors. What are the lessons that can be learned from these stressful conditions? For example, what can we learn with locally adapted animals? How can these be applied to our commercial systems? And looking towards breed substitutions and how new technologies such as CRISPR and gene editing can be used from the lessons we can learn from these other these stressful conditions, and how this can also help us to attain sustainable development goals for the United Nations. Therefore, how to need to increase sustainable animal production to meet global demands. So this is actually a picture from one of our speakers today, Dr. Veerasamy Sejian, from a paper that was published recently, looking at all the different aspects that can help in the adaptation of animals to heat stress. And as I said before, we have people from India, from South Africa, from Brazil, and the United States that are here with you today. So here are going to be speaking. First of it will be Dr. Veerasamy Sejian from India, speak on the comparative assessment of thermotolerance in different indigenous goat breeds. Dr. Michael Scholz from South Africa will look at the challenges and opportunities for climate smart beef production in Southern Africa. Professor Vinicius Fonsêca will look at thermal plasticity in dryland small ruminants to buffer negative impacts of climate change. And then Professor Harvey Blackburn looking at using genetic resources in the preparation for climate change. So our first speaker will be Professor Veerasamy Sejian from India, who works for the National Institute of Animal Nutrition and Physiology in Bangalore, India, looking at multiple stressors impacting small ruminant production in the changing climate scenario, identifying molecular markers for different environmental stressors in small ruminants, and primary focus on developing ecological zone-specific thermotolerant breed. Has 3-month international training on carbon trading and carbon sequestration and climate change in animal science at the Ohio State University. Had the Endeavour Research Fellowship with the Australian government to pursue a postdoctorate in the University of Queensland, and has received a prestigious Lal Bahadur Shastri Outstanding Young Scientist Award for his outstanding contribution in climate change and livestock production. Professor Michael Scholz from South Africa is the specialist researcher in applied animal breeding, extraordinary professor at the animal science at the University of the Free State, intimately involved in training of postgraduate students, professional member of the South African Council for the National Scientific Professions, member of the South African Society for Animal Science, where he was president from 2013 to 2017, looks at the improved production efficiency to reduce carbon footprint of beef, adaptation to global warming, counteract negative perception of carbon footprint of livestock production, represents the RC Livestock Group at the Global Research Alliance, participates on the UN Framework Convention on Climate Change, He's a rated scientist within the ANORF system since 2014. In 2017, received the Red Meat Research and Development Award for outstanding contribution to research, and recently received the gold medal from the South African Animal Science for outstanding contribution to South African beef industry. Congratulations for that, Mikhail. Vinicius Fonsêca from Brazil, visiting professor at the Animal Science postgraduate program at the Federal University of Paraíba, honorary research fellow at the University of Witwatersrand with the Brain Function Research Group, investigating mathematical modeling to estimate evaporative water loss in indigenous goats using miniature implantable biologgers. Postdoc position in the innovation group of biometeorology in Sao Paulo, looking at accuracy of methods to determine the cutaneous evaporated heat loss in livestock and testing the use of photovoltaic panels as the source of shading for animals. Looks at phenotypic behavioral and thermal adaptation to livestock and specializes in small ruminants raised in hot and dry environments. Methods employed to investigate the animal thermal equilibrium on housed and feed conditions and management of thermal environment to improve welfare of livestock. And finally, we're going to have Professor Harvey Blackburn from the United States, who's an animal geneticist, coordinator of the National Animal Germplasm Program for the USA, manages the development of acquisitions of animal germplasm for the NCGRP, coordinates 6 animal species committees that advises the NAGP about collection development, and since 1998 has been the USDA-ARS represented at the FAO's Intergovernmental Technical Working Group on Animal Genetic Resources, and was the committee's chairperson throughout the negotiation of the Global Plan of Action. So he's a real professional on that theme, which we had a long talk about yesterday, and is accepted by over 100 countries in the International Technical Conference on Animal Genetic Resources in the Interlaken, Switzerland agreement in 2007. So a lot of work, Harvey, and thank you very much for that. So after that's all over, after everybody does their presentations, then we'll have hopefully some time for questions. So if you have any questions, please put them up on the platform. And we will now start our presentation with our first speaker from India, as I said, Professor Veerasamy Sejian, who is going to talk to us today about comparative assessment of thermotolerance in different indigenous goat breeds. Thank you very much.
Thank you, Professor Koni. It's an honor to be in this platform to share some of our research findings. At the outset, I would like to thank the organizers for choosing one from India. I'm equally elated to listen to the other speakers cutting across the globe. This is the topic I have chosen to present today, comparative assessment of thermotolerance in different indigenous goat breeds. This is going to be the plan of talk for today. Concepts associated with different concepts on climate resilience in goats. Goat as the ideal climate resilient animal model, why we claim so, and then application of infrared thermal images for assessing the thermotolerance. And I will show some of the salient research findings, how to compare the assessment of thermotolerance across different indigenous goat breeds, and genomic approach to identify climate resilient animals. What is that rumen microbiome associated adaptive potential? I'll throw some light on that. And then I'll speak a little bit about all transcriptomics associated adaptive potential, and also a little bit on the epigenetic changes, particularly the DNA methylation pattern difference. Quantifying heat stress response in goats. And I would like to throw some light, as Professor Koni was projecting, strategies to sustain goat production in the changing climate scenario, with some conclusion and future perspectives. Well, this is a slide where it's a latest report 2021, where I've also been part. In fact, the research is from Chile. So where a couple of geographers have collaborated with us, and we have identified in a cluster based on principal component analysis and hierarchical clustering analysis into 3 categories. And you can see that India, it do not even fall in the most sensitive category, because it is falling outside India and Sudan, which is very alarming, because this is the scenario going to be prevailing by 2080. And you can see that Chile, most of the developed part of the world, comes in the cluster 3, where they are not going to be highly vulnerable. whereas most of the European countries and Asia and the African subcontinents are distributed in the cluster 1, where they are going to be highly vulnerable. They are going to face, the livestock population in these countries are going to face extremely severe heat stress for the number of days in by 2080 as well as 2050. Well, with that intention, I'm going to present my talk in different concepts. First, what are the different concepts associated with the climate resilient goat production? Well, you can see when you claim an animal to be climate resilient, it has to be adapting as well as producing optimally. So in that perspective, if you see, first is the adaptation, where there are different mechanisms like behavioral, physiological, blood biochemical, endocrine, cellular and molecular changes as well as morphological changes. The ultimate aim is to favor the animal to survive in a specific environment with a good welfare. Well, means the animal need to reproduce optimally. There are different endocrinological pathways the heat stress can affect through the negative feedback mechanism, as well as directly acting on the hypothalamus to reduce the gonadotrophin, and from the anterior pituitary, the gonadotrophin releasing hormone at the hypothalamus. So ultimately, there will be sequelae of event at the ovarian level, where the reduction in the estradiol, which can reduce the estrus expression. Ultimately, there will be reduction in the consumption and milk production. Well, of late, we are working a lot on the meat production characteristics, particularly both the productive as well as quality, meat quality variables, and some very interesting results I'm going to present in the later stages. Also, of late, we are also working on digestion process, how it gets affected, particularly the rumen microbial diversity also. These are some of the concepts, very important concepts, which can impart an animal the climate resilience capacity. To claim them to be climate resilient, they have to, apart from adapting, they should produce optimally as well. Well, this is what the concept of multiple stresses we have established in goat model. And you can see that this is a pictorial representation. The first part of the picture is a normal happening in an animal body, wherein the animals are blessed with some sort of body reserve. These are the productive function, growth, reproduction, milk, meat, and immunity. Whereas these are basal functions, which are very vital for survival. What happens in the if the animal is subjected to single stress, particularly if they are indigenous animal, they will be able to cope up to the single stress with the help of the body reserve without compromising any other productive function. What happens, suppose in Indian and African subcontinent, it's predominantly extensive system of rearing, where during the summer season there will be less feed available, there will be heat stress, and animal also to walk a long distance in search of this limited pasture, which is called walking stress, and also there will be less water available. So all these stresses, the cumulative stresses, the animal need to cope up if it is an extensive system of rearing in particular, wherein if they are subjected to 2 or more stress, which is practically happening, this body reserve is not sufficient. Ultimately, the animal have to compromise the productive function. So in our experience, it is in this order: growth, reproduction, milk production, meat, and immunity gets compromised. So because animal have to keep intact the basal function, because that is very vital for survival. This is what it is, and we have developed this concept, and then we have estimated with the several parameters, and a lot of sound publications have emerged from this particular concept. Moving to the second concept, goat as the ideal climate resilient animal model. In last 6 months or so, there's concrete research finding which claims, you can see that this is by 4.5 representative concentration pathway these results are, and you can see that by the current scenario, by 2050 scenario and 2100 scenario, goat is the least number of days the heat stress it is going to face by any means. You can see, like, but then the most vulnerable are going to be pig and the poultry, and then followed by dairy cattle and beef cattle, and also sheep. But then you can see that cutting across the zones by the 4.5 RCP, you can see that the goat is going to be the least affected. So that is the reason. This is by a group of researchers in the East Africa. And this is as a global, wherein we are also part of this collaboration. And you can see this is a global projection, where again you can see that goat is going to be the least affected for the entire globe as a whole, as compared to that of cattle, pig, poultry, and as well as the sheep. Well, this is how you can see, like you can see that the tropical climate are most suited for the goat. Most of the population of the goat are distributed particularly in the African and Indian subcontinent. Predominant populations are. That is because they have the advantage of better thermotolerance, unique feeding behavior, and they are better drought tolerance, as well as potential to produce less methane per unit of feed consumed, and they have the ability to survive on low pasture, as well as better feed conversion efficiency, and they are also highly disease resistant. This is particularly in the tropical climate. climate, and very, very low initial investment for the goat farming. These are some of the advantages which makes them in the tropical climate oriented species in particular. Well, this is our own team, along with collaboration with Professor Vinicius. We have brought a very good review on the subject, why goat to be the ideal climate resilient animal model. You can see that behavioral plasticity, which I am sure is going to address, means they can the browsing behavior, and then they can also consider the shaft of the tree, and they can have the selective feeding of drought tolerant variety cactus particularly. And you can see that more 6 to 8 goat can have a shade in the grazing condition as compared to that of a single cattle. That is a huge advantage again. And then this is what the review we are talking about just a couple of months back. Goat as the ideal climate resilient animal model in tropical environment, revisiting advantage over the livestock species. And this is what I was talking about, the entire global projection, increasing importance of heat stress for cattle farming and the future global current scenario. And this is the third concept, application of infrared thermal images for assessing heat tolerance. IRT has a very vast, wide, varied applications in animal production. And you can see we have a setup to identify the climate resilience based on eye temperature, forehead temperature, and we also estimate their shoulder temperature, back temperature, and flank temperature, and also the front leg temperature particularly, because in the tropical climate they will be having very lengthier legs, and then that temperature also makes a lot of sense. This is a set of parameters we usually do in all the breeds to compare their climate resilience. And moving on to the next concept, most important for the talk, comparative assessment of thermotolerance in different indigenous goat breeds. And you can see that the phenotypic traits, particularly respiration rate afternoon, pulse rate afternoon, rectal temperature afternoon, and panting score as well as skin head temperature. And you can see predominantly what I would like to draw the attention of the house is that out of the 5 different breeds, all are indigenous, very well known for their survival in harsh climatic condition. So the idea is to test which is the best breed. This is the 5 breed chosen from the southern part of the India. And then you can see that predominantly these 2 breeds were showing much lower heat stress response for these phenotypic traits as compared to that of the other 3 breeds. And you can see this is in terms of behavioral plasticity, water intake, standing time, lying time, drinking frequency, defecating frequency, urinating frequency, rumination time, and feed intake. These are all the behavioral responses. Again, you could see like there is a very less impact on these 2 breeds, particularly Kanniyadu and Kodiyadu, as compared to that of the Osmanabadi, Malabari, and Salem Black. What I mean to say is that they are behaving, at the very high level of THI, that is 95.5 something THI when we program. It's a very extremely severe heat stress, but then these breeds still feel not that much pressure as compared to that of the other 3 breeds. And this is with respect to the meat characteristics. And you can see for all the 5 breeds, the major carcass traits, the pre-slaughter weight, loin eye area, hard carcass weight, all are affected. But in these 2 breeds, none of the major carcass characteristics got affected. Only the meat quality gets affected in this particular breed as compared to that of the Kodiyadu. So from this pictorial representation, we can say that out of several parameters, I'm just giving the glimpse only 2 parameters got altered in the Kodiyadu, means like it is also maintaining its meat production apart from adapting to the heat stress condition quite effectively. So this could be projected as the ideal climate resilient breed, Kodiyadu. And moving on to the body weight changes, if you can see, like these 3 animals lost the body weight of the 45-day duration study, whereas these 2 breed have gained, because after the 4th week they started gaining their body weight because they are adapting better, and then their feed intake resumes back to the normal, and then they gain. That's a very unique potential for claiming them to be climate resilience. So that's how the body condition also increases, and also the growth hormone status also is of higher as compared to that of the other 3 breeds. Now moving on to some of the blood variables, typical markers, hemoglobin and hematocrit value and cortisol, aldosterone, triiodothyronine, and thyroxine. You can see that again, if you can see like in these 2 breeds, the levels were much lower for all these characteristic variable, particularly T3, T4. If you can see that the levels are more because they are not reducing their metabolic heat production. Their metabolic activity is at its peak. They are not much relying on reducing the metabolic heat production. That is the reason the thyroid hormones were of higher level, whereas the cortisol level, you can see that they are significantly lower in these 2 breeds. So I mean to say like they are extremely adapted breed as compared to the other 5 breeds. So overall, out of these 5 breeds, we have identified Coordinadora to be the stand-apart breed, and Karnataka to a certain extent, Salem Black also showed the promise. What I wanted to show is that you see that from this single experiment concept, you can see that the quantum of publication that has emerged. It is not like we are going to show like we are published more, but then the concept has been accepted by wider publishers. Moving on to the next genomic approach to identify climate resilience, you can see that this is what currently our research group is working at Bangalore, India. You can see that we wanted to target some unique adaptive potential, means something having a novelty from whatever earlier reported. And so we are trying to target two organs, that is skin as well as the rumen microbes. So from the sweat gland means also we are measuring active sweat gland measurement. These are all the concepts we are developed, which can we are studying from the skin, the epigenetic changes, and from the skin again the genetic traits, particularly the transcriptomics based approach, and then from the rumen microbes, rumen microbial diversity we are establishing, which can at the molecular level can give epigenetic changes, DNA methylation pattern difference, histone molecule modification, non-coding RNA, and chromatin remodeling. As far as transcriptomics, sweat metabolomics, skin metagenomics, and the skin and liver transcriptomics approach we are following. And for the rumen metagenomics, both 16S RNA sequencing as well as whole metagenome, shotgun metagenome sequencing as well, which can identify the microbial diversity and also the different pathways that could alter the stress pathways. So ultimately, this can give an generation interval adaptive changes and the concrete unique adaptive potential and the rumen microbiome associated adaptive potential, which can establish climate resilience. This is the rumen microbiome associated, just an example of 3 different breeds, Westernabody, Malabari, and Salem Black. At different taxonomic level, if you can see, here it is 1 phyla, and it is in the class 6 and genus 2 microbes got altered, whereas in Malabari breed, it is 2 phyla, 7 class, and 4 genus, whereas in Salem Black breed, it is only 1 phyla, 2 class, and genus only 1. So which means like this breed is keeping intact its microbial diversity much better than the other 2 breed. And also you can see that at a different level for all the phyla, class, and genus, and also the Keck pathways and the cellular process, environmental modeling, and genetic remodeling information and metabolism and organismal system. This is all the metagenomic approach. You can see again they showed least variation in these 2 breeds, the Karnataka as well as the Coordinadora breed for the rumen metagenomics approach. And that is what from the rumen metagenomics findings, what we are trying to see is that each breed have a distinct group of set of microbes which impart them the adaptive potential, suggesting there are host and microbiome interaction in the rumen, which can contribute to the variation in heat stress response. That's where, based on that approach, we have selected Karnataka and Coordinadora breed to be the stand-apart breed from the metagenomics aspect. This is the whole transcriptomics associated adaptive potential assessment again, and you can see that this is the volcano plot for the 3 different breeds, Westernabody, Malabari, and Salem Black. By gross appearance, you can see that very less number of significantly up or down regulated transcript in the Salem Black breed as compared to that of the Westernabody and Malabari breed. Well, in terms of numbers, you can see that the differentially expressed genes, which is significantly very higher in the Coordinadora breed, which means it brings an at a different level of transcriptomics level, very, very high impacted effect, particularly on the genetic modification level or at the genetic level or at the molecular level. The response mechanism of Coordinadora, which all these responses were very, very subdued, but then the transcriptomics based approach was very highly activated in this breed, which imparts them the unique potential to adapt for that extremely severe heat stress. And likewise, the Keck pathway as well as the COG pathways also, you can see that very, very highly activated as compared to that of the other 4 breed, which makes them another summarized to a very important slide of my presentation. You can see like we know like black absorbs heat and the white reflects the heat. So we know like based on the coat color and then the sweating rate, we can program the differences in the adaptation. But then this is the first of its kind report. As I told, that skin we are taking, and you can see that the real difference at the transcriptomics level, if you can see, the Karnataka breed, which is the black breed, predominantly you can see that number of genes that got altered from the skin, and around 3,681 upregulated and 2,872 downregulated, in contrast to the Coordinadora breed uniquely, which is only 401 upregulated and 175 downregulated. That is a typical example of how a coat color alters the adaptive potential of a breed. So we got some at the transcriptomics level that real differences have been established in this particular study. And from this, what we summarize is that out of the 5 breeds, the Karnataka and Coordinadora were stand-apart breed. Even among them, these are the feed intake, body weight, water intake, hematocrit, hemoglobin, cortisol, triiodothyronine, thyroxine, and various heat shock protein genes were remaining intact across the breed, and they also retained their productivity, and they relied only on behavioral response to an extent physiological response, and only HSP 110 was found to be the molecular marker in both the breeds, whereas here standing time, lying time, drinking frequency, defecation frequency, rumination time, panting score, respiration rate, pulse rate, rectal temperature, as well as skin temperature were established to be the markers for both the breeds. And ultimately, if you can see, like maybe for this breed, even a quantum of heat stress is very highly needed for their normal performance. That's what we concluded. Does heat stress acts as a stimulant to both this breed? And you can see that real difference in terms of THI, 25.34 than the control. This is a climate chamber study, so it's a very controlled study with a 25.34 in the heat stress. It is 94.76 the THI, whereas 69 ideally the thermoneutral zone for the control animal. So this is a very concrete proof wherein both the breeds were taken apart. And from these two, because we went for the epigenetic changes study, wherein bisulfite sequencing we have done for the DNA methylation pattern identification, and we can see that overview of genomic methylation in 2 different indigenous breeds, that is Karnataka control, Karnataka heat stress, Coordinadora control, and Coordinadora heat stress. You can see it can be very negligible, but then it contributes to several million reads actually in terms of differences. And you can see these are the 3 predominant regions where MCG, MCH, and MCHH are the 3 predominant regions where the methylation usually takes place. And based on this, you cannot predict their adaptation, but then it is an indication where predominantly the methylation happen. You can see that the CPG region where the most of the methylation, this is the violin plot which describes them. And this is the differential methylation actually. Important slide to understand differential methylation pattern. I have already told in the CPG region, predominantly the methylation happened. From that, the differences, we can predict their adaptation. You can see Karnataka breed, the differentially methylated region was significantly higher as compared to that of the Coordinadora breed. Likewise, the number of differentially methylated genes are also where significantly higher in the Coordinadora breed, in the Karnataka breed as compared to the Coordinadora breed, which means what we can infer from this slide is that the Coordinadora breed, which was standing apart, which is maintaining the production, which is maintaining the meat production, meat quality characteristics, growth, and everything, and then they are also adapting uniquely. Even the DNA methylation pattern was significantly lower, indicating this could be the stand-apart breed, which could be claimed as climate resilient breed in the southern part of the India. And this is the pathways wherein I'm not going into the details across the breed. And this is the common pathway altered for both breeds, and this is for particularly the thyroid hormone pathway, and the metabolic pathway got altered in Karnataka breed to show that in comparatively this is a sensitive breed as compared to that of the Coordinadora breed for the heat stress, whereas mostly the metabolic pathway wherein their energy metabolic pathway, particularly they are channelizing both for the production as well as for the adaptive mechanism. That's why we claim Coordinadora breed to be the superior breed. And these are the different biomarkers based on different study. In our experimental finding, we have established them to be different markers, like in the lymph node toll-like receptor 1, 4, and 5. In the thyroid gland, the thyroid hormone receptor, respiration tract is a respiration rate, hair, slick hair gene, and from the blood, hemoglobin, PCV, cortisol, T3, T4, HSP, HSF1, interleukins, and NRMP1. And from the skeletal muscle, it is superoxide dismutase, nitrous oxide synthesized. There are several enzymes which we identified them to be marker from the liver, and from the skin, prolactin receptor gene and fibroblast growth factor have been identified. And we also done some association analysis with the different phenotypic and genotypic traits with the THI, temperature humidity index, and we have established respiration rate, rectal temperature, skin temperature, panting score, and sweating rate as the physiological trait which can be claimed as marker, and behavioral traits such as drinking frequency, feed intake, water intake, rumination time, and from the immune system point of view, toll-like receptor 2, 4, 8, interleukin 2, 6, interleukin 10, interleukin 18, and tumor necrosis factor alpha, interferon beta, and gamma. All the genes would be considered as immunological marker. And from growth related, growth hormone receptor, insulin like growth factor 1, and adiponectin. Non-invasively also we have established hair cortisol, saliva cortisol, fecal cortisol as well as urine cortisol, because this is what we need to do, because non-invasively we need to identify them, the markers. And thermotolerant genes, several heat shock protein genes such as 27, 40, 60, 70, 90, 110, and prolactin receptor and superoxide dismutase nitrous oxide synthase and thyroid hormone receptor genes, apart from heat shock factor 1 also. From the meat quality point of view, myostatin, leptin, calpain 1, calpain 2, calpastatin, and crystalline alpha protein, and then diacylglycerol transferase 1 is another genes for the meat quality reflecting biomarkers. And from the blood, hemoglobin, PCV, cortisol, aldosterone, T3, T4, and HSP70. These all be found to be ideal biomarkers for quantifying heat stress response in indigenous goat breeds. This is what the review. We have also genes for resilience to heat stress in small ruminant. And last concept, strategies to sustain goat production in the changing climate scenario. That's what Professor Koni have already mentioned. We are grouping them into 4 different pathways. And this could be these two, the animal housing and nutritional modification could be short-term approach, whereas the genetic and the health management could be long-term approach. Ultimately, the target is that providing the appropriate microclimate with appropriate ventilation, cooling devices, mist cooling, sprinklers, and then forced ventilation, and then east-west orientation, and then side walls should be kept open for the wind flow, and etc., etc. As far as the nutrition, season-specific feeding, fiber feeding, feeding more fats and concentrates, and vitamin and mineral supplementation, and water, ideal provide cool drinking water. By far, all the approaches of animal genetics and breeding, we have identified like the ultimate end is the identify more thermotolerant breeds to cope up to the future. That could be the way forward. And also it is a neglected part, the animal health management, because we don't link them, particularly the vector-borne diseases are more. We don't link them with the geographical information system that need to be done to have a linkage between the climate change and the disease occurrence, and that has to be followed with the laboratory and the field research. So this is what we are projecting, and I would like to conclude my talk by saying like the latest available literature projects goat as the ideal climate resilient animal model, and there are differences even among the indigenous breeds. And particularly from based on phenotypic to ultimate epigenetic changes, we have identified Coordinadora breed to be the stand-apart breed among the 5 breeds. And efforts are needed to identify indigenous climate-resilient goat breeds which have the ability to survive in different agroecological zone. From here, we wanted to see whether the Coordinadora breed, when we are shifting them to the different agroecological zone, whether they are able to survive and produce in a similar fashion. That is what the way forward for us. And metagenomics and transcriptomics approach definitely provides scope for identifying climate resilient livestock accurately. Bisulfite sequencing related NGS analysis can quantify the epigenetic changes associated with the thermotolerance in the goats, and also sustaining goat production in the changing climate scenario must target improving production efficiency through strategical intervention and to augment the goat production in the changing climate scenario. Well, these are the future perspectives, the last slide of my presentation, and you can see like we need to quantify more intensified heat load index, incorporating all the weather variable, because THI only takes into account the temperature and humidity, and also we need to strengthen the agro advisory services for the farmers to prepare themselves. And then nutrigenomics, like we need to have more of a nutrigenomics concept to identify more climate resilient animal, and also metagenomics, particularly the microbial diversity associated. We need to do more shotgun metagenomics at functional level. What are the real differences across the breed we have to identify, and also we have to quantify more in detail the epigenetic changes, stress pathways, and also we have to identify more advanced biological marker which could be incorporated in future breeding using marker assisted selection. And also we need to identify animal which could produce less methane through immunization approach. And most importantly, the climate smart livestock production is the way forward, because even if one enterprise fails, the farmer need to have an advantage with other enterprises. So it's an integrated farming device that need to be in place for the future for safeguarding the economy or the economic security of the poor and marginal farmers, particularly in the developing part of the world. Well, I'd like to say special thanks to the organizer of ALAC 2021. It's been a pleasure sharing some of our novel findings in this platform. And as always, Professor Koni McManus, who has been very supportive for me. We have been in touch for quite some years now, and then she is sort of a role model for us, for me and Professor Vinicius, because she guides us perfectly and passes on very good information information for us. In fact, she is the one who is instrumental in organizing this symposia, and she has invited personally me for this. A special thanks to Connie as well. And these are the funding sources: Indian Council of Agriculture Research and European Union, CIRAD, France, and Australian India Council, DFAT, Australia, are predominantly the funding source for me. And my acknowledgement are due to all my collaborators, my present director and future directors, as well as my international collaborators. Most importantly, my students, who are all my strength to generate this quality data with us. And a special thanks to all the students who are placed abroad internationally for the doctorate after pursuing masters with us. And thank you so much for the opportunity.
Thank you very much, Professor Sejian. It was an excellent presentation. I think lots and lots and lots of information, lots for us to think about. And I think your last slide is a perfect introduction for our next speaker when you talk about climate smart agriculture. And we're now going to go to South Africa, and we're going to talk to Professor Michael Schultz, who's going to talk about the challenges and opportunities for climate smart beef production in Southern Africa. And as I said before, Professor Schultz has been working in this area for a long time. He works in Irene in South Africa, just outside Johannesburg. Michael, it's with you. Can you open up your microphone, please?
Mute, okay.
Yeah, that's great.
Let me now see whether I can run my. Can you see it now?
Yes, but you have to put it on presentation mode.
Yes, let me do that.
Yeah, perfect.
And now you can see it, okay.
Perfect, thank you very much.
All right. First, I want to say thank you for the invitation, Connie. It's really a privilege and an honor to be part of the symposium. As you can see from this first slide, there are a lot of people that collaborated on this research, and I want to acknowledge them. My outline is I'm going to give a short background on climate change. Then I'm going to talk of the effect of climate change on beef production. I'm going to say something about genomic signals of selection, opportunities, and the way forward. So let's start. Climate change projections for Southern Africa are in agreement that the subcontinent will become warmer and drier. On the left-hand side, you have the temperature, and you can see it's going to increase. On the right-hand side, you have the rainfall. The darker areas are going to become drier, and there are small areas that may become wetter. So if we look at temperature, the projections shows an increase in average temperature of between 1.5 and 2 degrees Celsius. That's the prediction. But it will range from a half degree Celsius to 3 degrees Celsius in the eastern parts of Namibia and the western parts of Botswana, and more heat spells are projected. If we move to rainfall, the rainfall projections confirm a generally drier Southern African region, except for the central interior regions of the Eastern Cape, where a wetter future is projected. But we're going to get significant reductions in rainfall of up to 40 millimeters per annum in the eastern parts of Limpopo and in Mpumalanga, which are already dry areas, and more variable rainfall is also predicted. So let's now look at the effect of climate change on beef production. And I'm going to start with the direct effects. Factors such as temperature, solar radiation, humidity, and wind all have direct effects on livestock. Ambient temperature is the factor that has the largest direct effect on ruminants, and most ruminants are best performing at temperatures of between 4 and 24 degrees Celsius. In Southern Africa, we are going to exceed these temperatures. Therefore, it's important that we have beef cattle that are adapted to these higher temperatures. Let's look at the indirect effects. Factors such as digestibility of feed, quantity and quality of the grazing all have indirect effects on the animals. And as temperature increases, the vegetation might change from C3 to C4 grasses, and in some areas the grazing capacity is expected to decline because the rainfall will be lower, and this will result in natural pastures with both a lower nutritional value and a lower tiller density than in the current temperate regions. So what is the effect of heat stress on reproduction? The direct effect or the direct response on reproduction is linked to the alteration and impairment of the cellular functions of various tissues in the reproduction system. The indirect response is linked to the redistribution of blood flow between organs, reduced feed intake, and respiratory alkalosis. And I think this is somewhat in line with what the previous speaker presented. So what will happen? Bull fertility is negatively affected because semen quality decreases and sperm defects increase. Furthermore, oocyte growth is compromised by altering the hormonal dynamics. And lastly, in early pregnancy, embryo development is impaired and there is an increase in embryo mortality. And I'm going to discuss 3 studies that looked at the effect of heat stress on reproduction. Let me just first mention that semen quality decreases when bulls are exposed to high ambient temperatures. And after a period of heat stress, semen quality does not return to normal for approximately 8 weeks because of the length of the spermatic cycle. And that leads to a carryover effect. So it is important to note that only 1 day of heat stress may reduce semen quality and potential fertility. It is therefore important to use bulls from adapted breeds during periods of heat stress. And let me now go to the research study. The first study was conducted in the central bushveld region at the Roodeplaat Research Station, which is just north of Pretoria. This was done over a 7-year period, and approximately 100 Bonsmara cows were involved. A discomfort index was calculated monthly by taking both the month's temperature and relative humidity into account. And it was also indicated that a discomfort index above 90 was considered to be very uncomfortable, resulting in heat stress. Now let's look at the results. The highest calving percentage of 90% was obtained when the discomfort index did not rise above 89 within 2 months prior to the breeding season, during the breeding season, and after the breeding season. So when the discomfort index was below 90, the calving percentage was the highest. The lowest calving percentage of 61% was obtained when the discomfort index was above 90 within the month just before the breeding season and the first 2 months of the breeding season. And this may be an indication that heat stress prior to and within the first part of the breeding season had a negative impact on bull fertility. We also found that calving percentage had a high negative correlation with maximum relative humidity 1 month prior to the breeding season. So the higher the relative humidity before the breeding season, the lower the calving percentage was. Similarly, we had a lower negative correlation with minimum temperature within the last month of the 3-month breeding season. And this may indicate that the cows were unable to cool down during night in the warmer summer months, and that may have led to lower conception rates and resorptions. The second study was done at the Fallards Research Station, which is in the Kalahari, and the effect of temperature humidity index on fertility was evaluated over a 19-year period. This was done by determining the correlation between the average temperature humidity index during the preceding breeding season and the corresponding average intercalving period of the cows in the next calving season. And here are the results. You can see the average intercalving period varied from 373 days to 482 days. A high correlation of 0.64 was found between temperature humidity index and the intercalving period. This suggests that approximately 41% of the differences in the intercalving period could be explained by the variation in the temperature humidity index of the preceding breeding season. The last project looks on the effect of calving percentage of different genotypes. At the Forschungsinstitut Research Station, we have a crossbreeding project where Afrikaner, Bonsmara, and Nguni cows are crossbred with either Afrikaner, Bonsmara, and Nguni cows, or bulls, which we refer to as Sangha breeds, or with Angus and Simmental bulls. And I can just mention that 2015-16 was the driest and hottest year ever recorded in South Africa. I'm going to come to that later on, but this is what happened with the fertility during that very hot and dry season. The calving percentage of the Sangha sired heifers, that's indigenous adapted, was 79%, whereas the calving percentage of the Angus and Simmental sired calves were only 65%. This led to a new project where we are evaluating the in vitro crossbred oocyte and embryonic development under heat stress. And the specific objectives of this project is to determine the survival rates of bovine oocytes from diverse crossbred genotypes matured in vitro and incubated under heat stress. We are using two levels or two temperatures to mature these oocytes. We want to see whether there are breed additive effects on embryonic and development competency. And lastly, we want to determine if female embryos are more heat tolerant than male embryos, and for that we'll need embryo sexing. And then I want to move to the effect of climate on the pre and post-weaning growth performance. There are indications that heat stress in cows during late pregnancy can have downstream effects on the performance of the progeny during the suckling period, and that heat stress per se has a negatively effect on the growth of suckling calves. And here I'm also going to look at 3 studies. The first study was conducted at the Forschungsinstitut Research Station using the Bonsmara herd. And you can see the map indicates to you where the Forschungsinstitut Research Station in South Africa is. The weather patterns that we used were total precipitation, season length, and maximum temperature. In this graph, the red histogram is the average maximum temperature for the season, and the blue line is the environmental trend in weaning weight that was estimated from the blue values. And here you can see that the difference in seasonal maximum temperature between different years were almost 3 degrees Celsius. And we found the correlation between the environmental trend for weaning weight and temperature was minus 0.62. That means that 38% of the variation in the environmental trend for weaning weight could be explained by the maximum temperature of that particular season. The second study, we looked at the growth rate of the diverse genotypes from phyllodes. And as I mentioned earlier, the 2015-2016 season was the warmest and driest ever recorded in South Africa. During the previous 10 years, we had on average 1.9 heat waves with 6.5 heat wave days. In 2015-16, we had 12 heat waves totaling 71 heat wave days. So you can see it's 6.5 heat wave days on average compared to the 71 heat wave days. And it's interesting to see what the effect of this very hot and dry year were on the weaning weights of the different crosses. I've summarized the Sangha sires, the weaning weight, and the weaning weight of the British and European sires. And you can see during the 2015-16 season, the weaning weights of the two groups were exactly the same. The year later, when it was cooler and hot, cooler and more moisture, the weaning weight differed by 27 kilograms between the two groups. But most interestingly, the weaning weight of the Sanger sires, or the calves of the Sanger sires, only increased by 12 kilograms, whereas the weaning weight of the British and European sire calves increased by a total of 39 kilograms, showing you how big the effect of climate can be on the weaning weight of unadapted genotypes. We used the same animals at the same year, and we looked at the effect of heat waves on average daily gain. In the case of the Simmental and Angus types, the average daily gain decreased by 17% during periods of heat waves, whereas in the case of the Sanger types, the average daily gain only decreased by 9%. But what we also found, it seems that heat stress may affect meat color and tenderness in some breeds, but much more research is needed in this respect. Now I'm moving to something completely different, and that's genomic signals of selection. Studies on genomic signals of selection in South Africa emphasize the environmental adaptation of the Afrikaner and Brahman cattle breeds and the distribution of the different genes underlying signals of selection, categorized by their major functions, will now be presented in the next graphs. Connie, here is the picture that you also used last night. If we look at the Afrikaner, you will see that 70% of the genes identified were linked with function or adaptation. But none of the genes in the Afrikaner were linked to production. If we move to the Brahman, 46% of the genes identified were linked with function or adaptation and 11% with production. So it was surprising that there were no genes associated with production in the Afrikaner, because some breeders do select for production, and that only 11% of the genes in the Brahman could be linked with production. A possible explanation is that while breeders were selecting for growth, natural selection increased the frequency of the genes for function and adaptation to survive and reproduce under the harsh South African conditions. But we must also remember, in 1981 already, French observed that selection for growth rate in a stressful environment was achieved through increases in resistance to environmental stressors. So perhaps this is not so surprising. Now moving to opportunities. The first one I'm going to discuss is the use of indigenous or adapted breeds. I'm not going to say anything about the development of early warning systems or adapted feeding strategies. Then I'm going to look at alternative breeding objectives and alternative production systems. There are a large number of indigenous beef breeds in Southern Africa that can survive in the harsh local conditions. The South African environment necessitates beef breeding programs that makes use of adapted or indigenous genotypes, because these genotypes are productive and represent a rich complement of indigenous breeds. These breeds have proven to survive in harsh conditions, and they exhibit low susceptibility to diseases, and they are adapted to other environmental conditions. So the development of genotypes to adapt to climate change must be matched with the rate of climate change, and indigenous breeds will be easier matched to the rate of climate change since they are more ready, if I can call it that. Then I'm moving to alternative breeding objectives. And the first one is cow-calf efficiency for sustainable beef production systems. And Muchabi, like you saw earlier, is working on this aspect. The question is how to increase the weaning weight of calves in relation to a cow large stock unit, which is a measure of feed intake in our extensive beef production systems. And then we also need early in life indicator traits and selection criteria or breeding objectives to improve fertility in our extensive beef cattle herds. And genomics can play an important role in both these aspects. The second alternative breeding objective is selection for alternative measures of post-weaning efficiency. Here we will think about residual feed intake and residual daily gain, but perhaps a combination of feed intake and gain in a restricted selection index may be the most simple solution. Now I move to crossbreeding systems. And if we talk about terminal crossbreeding, any system with large feeders from small breeding cows will be more efficient because small cows eat less than large cows. We found in South Africa that the potential increase in weaning weight can be up to 46% exposed per cow exposed to mating, while the feed intake requirements will only increase by 1%. And these are some of the results that we found in South Africa. When the Simmentaler bull is crossed to the indigenous Afrikaner cow, cow productivity increased by 15%. Angus bull or the indigenous Nguni cow productivity increased by 21%. But if we use the F1 Afrikaner cow, it can increase to up to 49%. Charolais cross of the Afrikaner led to a 27% increase in the value of the meat produced, and also 27% less feed was consumed from weaning to harvest. Then in farmlands, we're also looking at a crisscross system between the indigenous breeds, where we cross the Afrikaner, Montmara, and Nguni in a crisscross system. And this seems to be a viable solution for our communal and small-scale sectors, but more research is needed in this regard. So what is the way forward? Climate change and higher levels of heat stress is a reality, especially in the Southern African region, where the observed rate of warming is about double the global rate. So matching of genotypes and production systems to the production environment will become essential, and the use of biotechnology and genomics would facilitate rapid selection for adapted genotypes. The combination of sources of excellence to conduct research and development in climate-smart livestock production is essential. Therefore, we need virtual centers of excellence, whether they are national, regional, or international. And with that, I'm ending my speech. Thank you very much.
Thank you very much, Michael. I think you showed us a totally different picture from what's going on in India. You look more at the from a production point of view rather than the experimental point of view. So I think, but looking at much the same techniques, but from a different perspective, I think that was really good. So now we're going to go to Brazil, and we're going to see the presentation from Professor Vinicius Fonsêca, who's going to talk about thermal plasticity in dryland small ruminants to buffer negative impacts of climate change.
Hi, everyone. Thank you very much, Professor Connie, for inviting me. It's an honor to attend this Congress. Today I'm going to present some aspects involved in the thermoplasticity in dryland small ruminants to buffer negative impacts of climate change, and how we can use biologging to investigate animals under free-ranging conditions. But first, the plan of my presentations will be talk a little bit about equatorial semi-arid regions and their stressors, cost of thermo-homeostasis of small ruminant, with emphasis on water and energy, climate change and its impacts for thermo-homeostasis, aspects involving in the thermoplasticity, employing phenotypic plasticity to regulate body temperature. Some examples in free-living ruminants. Phenotypic plasticity in farm is more ruminant. Why is it important? And lastly, take-home message. So first, here we are close to the equator, and in the Brazilian northeast, big part is the semi-arid, and some small parts are already arid environment, which means that animals faces frequently with high levels of solar radiations over the entire year. Very different patterns when you see some environments in temperate regions. So animals are faced with high levels of solar radiation and consequently high levels of air temperature and high levels of radiant mean temperature. Plus, they are facing, especially the small ruminant, which are raised on the pasture-based system or on free-ranging conditions, they are facing with very unpredictable and high fluctuations in the water and food. When in the Caatinga biome is one of the most important biome in the Brazilian Northeast, we can see different patterns and different periods of drought and short periods of rain. So the animals in evolution of animals, specifically of small ruminant in these regions, make this to have a suitable phenotype to cope with these challenges. But I must emphasize, firstly, the body characteristics as a higher surface area to volume ratio. It's more body size and they are very pure insulated. These characteristics will determine how these animals exchange heat and mass with its environment, and then how they implement behavioral and physiological responses to cope with thermal challenges. What we are doing here is to study more how these animals respond to the thermal conditions normally observing in the semi-arid regions. In this study, we evaluated animals by using a spirometric system to quantify the thermal equilibrium over the 24 hours. And what we found when we expose an animal to direct solar radiation or protected against the solar radiation, in this situation, in these extreme situations, the animal thermal equilibrium will be faced with from 9 to 2 o'clock. These animals will be faced with high levels of radiant mean temperature and high levels of solar radiation. And in terms of thermal equilibrium, how the animals cope with these conditions. As you can see, we quantify the thermal balance of this small ruminant sheep breed. They call it Moradonova. And what we observed during this time, that is the energy that this animal gain by sensible heat. The amount of heat coming in from sensible means it's 350 plus metabolism of close to 40 and a respiratory evaporation for 15. Under such conditions, the animals will accumulate a high amount of thermal energy, which will require for them to dissipate a large amount of heat by evaporating water from the skin. But when the animals are facing with these conditions in free-ranging condition, actually, they prefer to seek shade and avoid to use a lot of water to lose heat. As in this type of environment, the water shortage is a very common environmental stressor. In our positive situation, as these animals are very poor insulated, when we look for the thermal balance of these animals during the nocturnal period, and when we plot the metabolism and the sensible heat loss against the air temperature, average of air temperature, what we can see that on the temperature below to 24 or 23, the sensible heat loss must be exceeding the heat produced by metabolism. So these animals will be facing with difficulty to conserve heat. And what they do normally under free-ranging conditions is to employ the huddling behavior, a behavior that helps animals to conserve heat and reduce requirements to increase the metabolic heat production, as you can see here. So in all of these experiments, animals were hydrated and animals were non-fasted, in a non-fasted condition. But as we explained, a normal situation faced by these animals in an equatorial semi-arid region is the heat challenge coupled with the water shortage and energy shortage. In this condition, the cost of thermal homeostasis in terms of water and energy will be high for these animals. And the impact of the climate change will therefore impact negatively the maintenance of thermal homeostasis, especially when the thermal challenges are combined with the water stress and energy stress. So the predictions of the climate changing is that the climate in this region specifically will be drier, and under such conditions, how to cope with these rapid changes. So one important thing when we are looking for the impact of climate change on animal performance is that how fast the events of climate change is progressing and how these will impact the capacity of animal population to employ genotypic adaptation. In a recent study, as we can see that the current rate of climate change is progressing fast and can impair genetic adaptation of small ruminant and semi-arid areas. In conditions like when animals are facing with thermal stress, water and energy stress, what they normally do based on studies performed in the arid environment with species adapted to this region, these animals face it also with seasonal environment. And when they are a viability of water and energy, they are able to regulate the body temperature relatively constant. But when the water and energy are suppressed on the environment, these animals employ his phenotypic plasticity to thermoregulation by decreasing the resting metabolic rate and suppressing evaporative cooling means. To do that, we need to evaluate animals in free-ranging conditions. And the only means to do is by using biologging. It's the implantable and miniature devices from which we can measure the microclimate selection, activity, metabolism by using the heart rate as a proxy of metabolism of animals in free-ranging conditions, and the patterns of body temperature by using the deep body temperature as in the intraperitoneal cavity and in subcutaneous cavity. So by using these two body temperature, we can see the vasomotor state changes of animals when they need to be more vasoconstricted or when they need to be more vasodilated. A simple example of the studies performed with the free-living ruminants in Africa, more specifically in the Kalahari Desert, by using some of these biologgers. So they are very easy to implant on animals. And what they have as a special feature is that we can overcome the limitations of the transmission of data in terms of distance, and we can evaluate animals for a long-lasting time, like for 1 or 2 or 3 years continuously. So, yeah, this is a picture of implantations in biology in a goat that I did in South Africa. And here is one study explaining how oryx, Habsburg, employ the phenotypic plasticity by using adjustments on their behavior to save water in the periods when they are facing with heat and water stress. Results by using implantable accelerometers showing that during a period when we have heat challenge but wet period, these animals, distribute activities over the period with more early grazing patterns in early morning and at the end of the day. But when these animals are facing with heat and drought, or in other words, with heat and water stress, what we can see is a complete change on activity pattern. We can see the nocturnal grazing pattern of these animals, which help them to save water. So during the day, these animals prefer to be under shade. That is the best way to manipulate heat exchanges and decrease the heat load absorbed by the radiant environment. The other study, what we performed 2 years ago that we did with wildebeest. So in this study, we evaluated these animals over the continuous 3 years. And during this period, we implanted some biologgers in the ruminal cavity to measure the heart rate and the ruminal temperature, and also the biologgers to measure the body temperature. So now I will show you how the animals change their metabolic responses based on changes on precipitation and in the availability of energy in the environment. So by using this biologin, we can see that precipitations pulses are responsible to change the metabolic heat production by using the heart rate as a proxy of metabolic heat production and activity. So this wildebeest, the pieces of precipitation determine activity and metabolic change of these animals under free-ranging conditions. So at our goats in semi-arid, what is the main aspects involving in his capacity to employ phenotypic plasticity to buffer negative impacts of climate change? Based on this, what we know about that is that the phenotypic plasticity of small ruminants living in equatorial semi-arid region are very poor investigated, especially in conditions where animals are free-ranging. Models predicting animal responses to climate change and lacks impacts of multiple stressors for free-ranging livestock. And other motivation is that models predicting animal responses to climate change do not take into account capacity of animals to employ phenotypic plasticity. Based on this, we would like to address these questions. Are free-ranging goats employing thermophenotypic plasticity in an equatorial semi-arid biotop? Now we just implanted in 20 animals from Moshoto goats breed and Canidate goats breed. We implanted biologgers in animals to measure several responses that will indicate us how these animals will employ this thermophenotypic plasticity to regulate the body temperature. Based on this, what we are looking for is to see the impact of thermal challenges, water stress, energy stress on how these animals will respond during 2 years continuous study. And to do that, here is the incision, what we implanted, a logger to measure activity, a logger to measure body temperature in an intraperitoneal cavity, and a logger to measure the subcutaneous temperature. And also we put a collar, where in this collar we have a data logger to store data that is coming from a ruminal unit that we use to measure the ruminal temperature and heart rate. So together with measurements of body temperature, activity, microclimate selection, and heart rate, periodically we will also measure the blood stress biomarkers to see how the multiple stress factors impact the growth performance, how animals will use some heat conservation or dissipation strategies, and how the animals also will use some water and energy conservation mechanisms to cope with multiple stress factors in these areas. But firstly, we just finished our pilot study to see if these devices will impact in some way the normal behavior of animals under free-ranging conditions. And the preliminary results of this pilot study, what we did, we implanted in 4 animals the devices and evaluated in other 4 animals without these devices. And what we observe in terms of normal pattern of behavior is that the collar or the implants do not change any pattern of the grazing behavior over the day. Also, the bipedal stance behavior, which is very important as a behavior to show how these animals are plastic to seek food in some parts of the Caatinga, which other animals are not able to do. We can see that the presence of collar or the implants did not impact the expression of this behavior. And finally, looking at the 20 continuous days of measurements of body temperature and with the measurements of heat load experienced by these animals, because on the collar, the animals have 2 temperature sensor, which will measure the temperature where the animals is instantaneously. So what we can see in this picture is that the high sensitivity of the sensors in 20 days, when we have more hotter days, the animals were highly vasoconstricted. As you can see, the subcutaneous temperature much lower than the intraperitoneal temperature here. In a positive situation, in more cooler days, in more hotter days, the animals could be highly vasodilated. So this type of biologgers are suitable to evaluate the capacity of animals to employ the phenotypic plasticity with high sensitivity and accuracy. So my take-home message is that the direct impact and indirect impact of climate change will increase the conflict between homeostatic system in free-ranging ruminants. The use of miniaturized animal implantable devices for logging data over extended period of time will elucidate if a small ruminant employs some type of thermophenotypic plasticity in response to heat load, lack of food and water. And understanding the extent of this plasticity will help us identify resilient breeds of sheep and goats and better predict their potential to adapt to the rapid change in climate. I would like to thank you very much to my research team. And I would like to also thank you very much for Professor Connie for inviting me and all the participants. Thank you very much.
Thank you very much, Professor Vinicius. A very interesting talk, looking at practical conditions in the Caatinga region of northern Brazil, and also making the connection with South Africa. I'm sure Michaela will have lots to say about that later on. Interesting that they have this overlap in the connections between the 2 countries.
I took pictures of his presentation.
I'm sure he can send you it later on if you want.
I will contact him.
We now go to the United States, and we talk to Professor Harvey Blackburn from the USDA, who's going to talk about the work that they're doing there on the use of genetic resources to prepare for climate change. Harvey, turn on your microphone.
Yep, I just did. Can you? Thank you very much, Connie, and thank you for putting this together. It's been a very interesting discussion that we've had this morning or this evening, depending on where you are in the world. What I'd like to do is just give you an idea of some of the things that we've been working on in relation to looking at genetic diversity and climate change. There we go. I think, as Connie mentioned earlier, and obviously different speakers today have talked about, there's a number of factors that we need to be concerned about in terms of climate change, heat stress. We've talked about the increase of diseases that we may or may not have had prior to these kind of changes. We also have to deal with issues like reduced forage quantity or quality, and maybe the need for greater mobility of our livestock. I think that when we look at climate change, why a lot of emphasis, it seems, has been placed on talking about different breed types, different phenotypes that may exist, and the possible suggestion that there needs to be various changes in the types of breeds that we use in preparation for climate change. But as I think Connie mentioned in her introductory slide, you know, there's a whole range of characteristics that we have the opportunity to really look at, employ, and to change essentially our animal populations to meet this challenge. For example, body shape, body size, why in the United States, why we've dramatically changed body size in our livestock. If you go back to the 1960s, we had cattle that were maybe waist high. In the 90s, why we selected for extremely tall, long cattle, cattle that were maybe approaching 6 feet or not quite 6, but close to a little over 5 feet. And I'm just trying to think that'd be about a meter and a half in height in the metric system, and discovered that we didn't like those cattle, and the cattle we have today are shorter, but they're also extremely thick-bodied cattle. And of course, as we've heard from earlier speakers today, why you know, we have to have animals that are going to be able to shed heat under these conditions. And so certainly changing that body size and shape is one factor, and a factor that we know that we can do quite easily. If anything is evident in the United States, when you go to the southern part of the United States, you see increased use of Bos indicus cattle. What strikes the major difference there is you start seeing, okay, these cattle have larger ears, they have more brisket or sheath on the brisket, their sheath, the penis is longer. All that lends itself to cooling. And so clearly, you know, we can, you know, and we've spent years trying to get rid of that, particularly among Bos indicus, but clearly it's something, you know, that we can look at in terms of trying to alter in our Bos taurus breeds as well. Of course, hair density and type is another. Looking at other observable phenotypes, a number of ways, the Famancia score for parasite load. Of course, we've talked a lot today about complexity, measurements that take a lot more technology, hormonal profiles, genomics, GWAS, or fine structure, which is what I'd like to visit about today. But you can see, I think, from these two pictures, the opportunities actually that we have. That calf in the bottom there is actually a Holstein, but we've integrated the slick-haired gene into that Holstein. This was done down at the University of Florida. And so by that simple integration, why we've better acclimatized Holstein, particularly those produced in the southern United States at this point in time. So we have a lot of flexibility here, and there are many options or routes that we can take to modify our populations. What intrigues me is that we can, of course, use integration, crossbreeding, various populations. But when we start looking across the literature at the variation that we see, and these are mainly from molecular studies looking at genetic diversity, why we see that the largest proportion of genetic variability is actually found within our breeds versus between breeds. That's very consistent throughout the literature in any part of the world, suggesting that we have a lot of variability within a breed to make changes that we feel are necessary for animals to remain productive. So what I'd like to do is, taking that approach, is look and show you some of the results that we've had as we look at a couple of different studies using Herefords. Hereford for us is a interesting model, if you will, for breeds, because one, it's widely distributed across the United States. As we go into different ecoregions, why we can almost always find Hereford in any production system. The first study was some work we did with some of our Brazilian colleagues in 2017, looking at more of a fine structure analysis. We using about 14,000 SNP markers and using a subpanel of 66 there. As we'll point out, why we started with a fairly large number and that was reduced. We'll talk about that when we look at the next slide. The fine structure that we did was using the 66 SNP markers that are associated with heat shock proteins and production characteristics. The other was a follow-on study where we looked at the entire pedigree of the Hereford breed. So we're dealing with a pedigree file of over 200,000 animals, and using random regression as a method to evaluate, and we used as a proxy for adaptation to climate as intermuscular fat was the trait that was used as a measurement. And in that study, why, as we'll see, population was partitioned by longitude and latitude, and then of course using heritability as a measurement of comparison for IMF, and that well-used measure in terms of looking at genetic bioenvironmental interactions. For the first study, why we started with a very crude map, and later on you'll see how far we've advanced with the help of Dr. McManus. But we broke the country, the United States, up into 5 different ecoregions, and you can see cool, arid, cool, humid. This transition zone, it never gets terribly cold, or it does get somewhat hot there, but the forage conditions are very different in that region. The warm, humid areas of the United States, which we see down along the Gulf Coast, and then the warm, arid regions. Now, what we did is looking at the SNP data that we had, the animals, where those SNP data came from, and this was, I think, a very key element in this analysis. We had to go in to the pedigrees of these animals and establish how many generations they had been within each of these ecoregions. So we only used animals that had been in a particular ecoregions for at least 2 generations, if not 3. So there was an opportunity for selection to work in terms of adjusting these animals to the particular environments that they had. And when we looked at the genetic migration or gene flow among regions, why that's what these gray arrows are. The size of the arrow indicates the amount of gene flow that we have between those regions. And you can see, like, from warm humid to cool humid, there's very little migration going on there. Transition zone, we have more back and forth between all the different regions there. And of course, when you come out to the cool arid, warm arid, why, again, very little gene flow is occurring between there. And we computed the coefficient of relationship between these regions. And you can see by this lower diagonal here that we have a relatively low relationship among the groups. So we achieved our goal, I think, of getting the cattle properly assigned to the regions that where they had been raised, and natural selection principally would have had an impact then on altering the allele frequencies for those populations. Just to give you an idea of, this is a graph of the thermal heat index for each of those regions. And the black line you see here is actually where if the THI goes above that, you're actually, the animals are in danger of having an extreme heat exposure. And so you can see, particularly in our warm, humid, warm, arid regions, that we're really talking about 4.5 months of time there where those cattle have the opportunity or potential to come under a lot of heat stress. So particularly when you compare that with, like, the cool humid or cool arid regions. So to look at our analysis, I started with using admixture to see if we could differentiate between populations as they would be assigned by the ecoregions. Here you can see that clearly there's large portions of cool, humid, cool, arid that fall into this first cluster. Obviously, the warm arid here, the big portion of its genome is assigned to a different cluster by itself, and then of course warm humid, while you can see essentially mirrors kind of this K1 and the K6 mirror each other as you go from the cool zones to the warm zones. So this gave us a lot of confidence that yes, we're on the right track. There are differences among the populations or subpopulations of Hereford as you moved to different areas of the country. Of course, we were most interested in looking at when you reduce that 14K panel down to just using 66 SNPs that are associated with heat shock proteins and a few production characteristics like milk yield, why we see in this principal component analysis the breakout of the zones, and they remain quite distinctive. You've got the warm humid down here, cool, humid up here. And then, of course, on the other side of this red line is the arid regions, the cool above the blue line, the warm below. And then interestingly for us, while we found this transition zone to be located on this midline to be quite interesting as well, in terms of saying that, okay, these cattle are in a different type of environment, that they may have a lot of opportunities to move into different regions if that was desirable. The next issue that or aspect of this study that we evaluated was to look actually at allele frequency differences for our panel of 66, and what we've got listed is those allele frequencies that were actually different from one another. I draw your attention to this upper left-hand corner. You can see that kind of oblong circle that we've identified. Those are two heat shock proteins. And actually what it's showing is that as you look at the warm humid versus the warm arid, there's a very distinct difference between them for both those heat shock proteins. Indicating that as you go into different types of regions, whether it's warm arid or warm humid, there could be totally different selection criteria or mechanisms that are in play, and those allele frequencies are going to be adjusting as you move into those regions to whichever way is potentially more important for the animal in that condition. So to take that a step further, why we wanted to know, well, okay, we see these differences in Hereford subpopulations within the United States, but how do they really compare when you switch over to looking at a breed that's more adapted to subtropical type environments like the Nelore cattle? And these were actually samples of Nelore that came from Brazil. And so when you run the same type of analysis, these are the same two heat shock proteins that we saw in the previous figure. And you can see that in this instance, the warm humid Hereford is quite close to the Nelore in terms of the frequency of the alleles at that loci. You see a reverse trend when you come to looking at the warm arid is actually closer to the Nelore. But either way, I think it's making the point that these cattle, the Hereford cattle, in these two harsher environments, are actually, their allele frequencies are moving in the same direction or toward a breed that has a lot more potential resistance to heat stress than what we would think of the Bos taurus. You can see when you go over to these other SNPs that these are all dealing with milk yield, how different they are from the Nelore. So that also kind of reinforces the idea that, yes, within particular SNPs, we can see these patterns start to emerge, and offers us an opportunity that we can use in selecting various cattle types that we want to have perform in a specific environment. So just kind of in summary, why the picture there, you give you an idea of what it's like out in West Texas, the warm arid region that we've identified. Clearly, we feel there are subpopulations that have been formed within the Hereford breed. The allele frequencies of SNPs associated with environmental adaptability, in this case heat shock proteins, have changed, most likely under the forces of natural selection, or potentially some correlated response with production characteristics as well. Just briefly, the second study was looking at all of the pedigree file of the Hereford Association, and you can see that based on color, why we've divided these cattle, assigned them to either groups by latitude or by the longitude. And so that was the basis for the analysis there and running the random regression approach. As I said, when you start looking at, we used heritability of intermuscular fat, and you can see that these estimates, and we actually broke the north into two different groups. South is in two different groups here, but you can see that as we move along the degrees of longitude, those heritabilities are changing. They're not consistent. And if we look at most of our breed associations, why they use one particular measure of heritability for a specific trait, and this really suggests that, particularly in the terms of climate change, that we should really be altering that and using heritabilities in our estimation of breeding values for specific environments and not count on a generalized heritability for a trait for an entire country. So this was the main feature I wanted to show you on those. Random regression allowed us to actually parse these differences out and show that there's really large and substantial genetic bioenvironmental interactions going on. And I think it further suggests that the existence of subpopulations and alterations in the amount of genetic variability that we have to use. Turning to kind of our main thrust here in Fort Collins and gene banking. So we're charged with collecting genetic diversity for all of our breeds of livestock in the United States. And initially, when we started this effort, why we looked toward broadly sampling breeds, populations within breeds, but not really with an eye toward looking at this question of collecting germplasm from subpopulations by ecoregions. And from our standpoint, this is critically important that we begin to do that and to understand these differences as well, because with climate change scenarios, why obviously we're going to have to be utilizing our genetic variability much more extensively maybe than what we have thought of in the past. Give you an idea of where we are at this point in time, some work that we've done looking at THI and putting that, making essentially new zones. The original map I showed you was just kind of what we thought as a general estimate, but this is a much more precise view of how the country differs by THI. The red dots that you can see there are essentially all of our beef cattle collection. And you can see that we've actually started to acquire germplasm from the various zones, some quite intensively. When you look in places like Nebraska or Iowa here. But that said, you can see as well that we've got an accumulation of animal germplasm in the repository coming from really some of the higher areas, areas that have higher THI than other parts of the country. When we look at the map, it also kind of helps us to identify gaps in the collection. So as you can see here, why when we come out to this part of the western part of the United States, why we're really missing a lot of samples from that region as well. So in conclusion, some points that I'd like to leave you with. Certainly from our view, why breeds within a country may already harbor useful allelic combinations, and that those frequencies may have been substantially changed over time. And so we need to be better quantify those types of differences that exist. From our view as well, breeders can employ within breed genetic variability to adapt animals to climate change. Large-scale breed substitution may not be needed for a country like the United States. Further evaluation of targeted genes and their associated SNPs may yield faster progress than other approaches. Thinking of the comparison between, say, our fine structure approach here and maybe GWAS, using GWAS methodology. And as I've ended up there, why gene bank managers need to consider environmental differences in collection development. Thank you very much.
Thank you very much, Dr. Harvey Blackburn. I think that's very interesting, looking at the on a country scale and looking at different environments. We actually mapped breeding values in Brazil for milk production, and we also saw regional differences related to environmental factors. So I think that it's, as you know, it's something that we're very, very interested in. So just to start off the discussions, we have around about 15 minutes. So maybe if we can go back to Professor Sejian and start off. You showed us an enormous amount of traits, an enormous amount of very relevant towards the question of climate change and adaptation of animals. And so, but I would like to ask, how do we define what is a desirable genotype under these conditions, and what sort of traits should animal breeders be looking to measure on a more practical basis? Because if we look at all of what is available, we sort of resume that or summarize that into something that could actually be useful for the animal breeding associations. And please, anybody else wants to join in the discussion, please feel welcome.
Thank you, Professor Koni. So I would predominantly suggest the shade-seeking behavior and the coat color, because if you see, all these are very well adapted breed, whatever we have chosen. In fact, there are other 3 breeds as well we have compared. Just for the paucity of time, I have chosen the best 5 breeds to compare themselves. And then you can see their leg length, and then their coat color, and then their shade-seeking behavior particularly, because when we go to procure this animal, we ensure that these populations are grazing during that time, and then we pick them from different location to have the heterogeneity of the population, because when you conduct a genetic study, the population need to be heterogeneous. So that's where we see whether the individuals are, because within, as the last speaker was telling, that there are within breed variations as well. When we pick these animals, we go to the grazing condition and we pick them where the animals are quite grazing without any individual variation. So those type of animals we pick, and then we see predominantly the shade-seeking behavior to be one of the important criteria, especially in the grazing condition, where we can select these animals. We know that they are all indigenous, and then that is the reason we wanted to, because our Honorable Prime Minister calls for doubling the farmers' income. Goats are tipped to be very low investment required enterprises for the livestock. So that is the reason we do target on this goat population, where it is a gaining momentum in India, where they can be given very good alternate livelihood security for the farmers. So that we look for predominantly their coat color, because we have also seen predominantly out of the 7-8 breeds we screen, the black coat color breed also performed equally in par with the lighter coat color as well. But then their leg lengths were very drastic difference there, because the white coat color breeds had the longer length of a leg than the black coat color breeds. So that summarizes in the field condition how to choose them. So and then we have reciprocated that with a different level of biomarkers assessment, right? The amalgamation of phenotypic as well as genotypic traits, and predominantly we have programmed them based on transcriptomics approach, and then we have validated the result with the Hitchcock factor 1 and HSP27 and HSP110 to be ideal biomarkers if you claim that genetic marker if you wanted to see, and then prolactin receptor gene, and then thyroid hormone receptor. These are the stand apart in cutting across all the breed. We have established them to be the markers.
Okay, thank you very much. Does anybody else have a comment to make on that, or can I go to the next question?
Yeah, I do, Connie. I'm interested, in the case of our Indian goats, and what I wonder about in terms of the coat color, if there's differentiation between coat color and skin pigmentation color. In other words, when we go to, for example, Brahman or Nellore, light hair color, but they're almost always very dark skin pigmented. When you go to Southern Africa, you've got the Swana goat that tends to be a white-haired goat with black pigment. Do we know whether, okay, you can have white pigmentation with a white coat color, but with light pigmentation? And do we know, are we getting, is it additive or is it multiplicative to the animal's advantage to have both of those characteristics, or do you see any difference at all with skin pigmentation?
We actually saw some things like that with sheep here in Brazil, because we looked at, we had hair sheep and wool sheep. With most of the hair sheep had darker color skin, but some of the wool sheep, for example, I think it was the Hampshire Down, was a white wool sheep with a darker skin, and it was seen to be more tolerant than the white sheep with the white skin. So, but whether it's additive or multiple, I don't know. I couldn't say that.
I just have a comment about that. Similar situations when we look for the Bedouin goats, nowadays we never were able to explain why the Bedouin people prefer, why the populations of black Bedouin goats are bigger than the white ones. Here, the case of Moradonova, the brown one, and the white one, hair coat, during the evolution process, I think the allele frequency for the hair coat, white hair coat, cover under the skin, non-pigmented skin. And when you see the interaction between the different populations of Holstein cattle, even here in Brazil, in the Northeast, I think the natural or directed selections of breeders prefer to use the Holstein cows covered by a black coat over the Holstein cow that has white spots, because the white spot are on the skin with non-pigmented, under a non-pigmented skin. I think the technology of gene editing, looking for a good combination between the expression of the characteristics that is really valuable in for example, in situations where the solar radiation it's a problem, like in our region here. Skin color, it's really important. Skin color, a black skin color, it's really important. So the gene frequency for skin color, black skin will be selected, and the correlation and the genotype or phenotype correlation with the hair color we do not know much about that. And yeah, and I think we should move forward to understand more how the gene expression aspects will determine the coat color and the skin color of animals. And I think the gene editing, it's a good way to find fast modifications in cattle population, depending on the environment, aspects. Yeah.
Thank you. Oh, I have a question for Michael, please. Here, congratulations to all the speakers. Excellent data presented. The question is for everybody, but I think Michael his presentation was more directed towards this, so I'll start with him. Considering that research to increase productivity is still important and a major driver worldwide, especially for developing countries that have large hunger issues, how do you balance climate change, the climate change agenda, with traditional animal breeding goals? What we have to keep in mind the problem that most local or indigenous breeds have not even properly been inserted within a breeding program. So I know you worked somewhat with that in South Africa. So
yes, thank you. I think the two issues are not contrasting each other. If we can increase productivity, we will also address the negative environmental impact, because we are going to increase the production per unit. And we looked at our indigenous breeds, and in the case of the Afrikaner, we looked at what happened with weaning weight, with fertility, and with cow weight. And the interesting thing is, over the last 30 years, the cow weight decreased, the calf weight increased, and the fertility increased. And if we use the tier 2 assumptions from climate change, it indicates that the environmental impact of the breed decreased by 12% over the 30-year period. So the important thing is to measure the correct things, and not to realize that maximum production is not always the most beneficial from a productivity and environmental point of view. So we are trying to balance that. And then, of course, selection for something like a residual feed intake, where you increase or you keep the growth rate constant, but you decrease the feed requirements, where especially in Southern Africa, where we don't know how long we will still have the luxury to feed maize to livestock. It's important to reduce the feed requirements while maintaining the production, and it's also linked to a lower carbon footprint. Thank you.
Thank you.
I completely agree with Michael, because that's where the challenge is going to be, because especially when the pasture lands are shrinking, and we are going to face a huge problem, especially looking into the fodder point of view. I think that is the reason, like most of the modelings are projecting the small ruminants to be the go-to species by anything after 2050 onwards. So that's very important. As you told, RFI, residual feed intake, is going to be one of the important criteria for genomic selection as well.
Okay, well, I have a question for Professor Blackburn. So the slick-haired locust was introgressed into the Holsteins. Was it by conventional breeding, or did you use other biotechnology methods?
That particular calf was conventional breeding. However, there are people that have been working on introgressing just the slick-haired gene using gene editing as an approach.
Yeah, so I think we're actually out of time. So I'm sure we have lots more discussions that we can have, but thank you very much. I think we've had an excellent morning, afternoon, and night, depending where you are in the world. And thank you for your very interesting talks. And I think we've covered a very wide, a very broad subject base on what's going on, looking towards animal production and climate change and what are the possible solutions that we have for these questions. And so thank you very much. And we hope to see you all again soon in person and not just through a computer screen. Okay.
Thank you very much.
Thank you, Connie. Thank you all. It's good to be in touch with all of you. Thank you.
Thank you.
Bye.
Bye-bye.
Everyone. Bye.